presenter, and learned participants. Very good afternoon, all of you. It is my pleasure to be here as a moderator of the today's webinar. So as part of the celebration of the 20th anniversary of AFOMP, we are going to deliver today the 11th webinar. The title of the webinar is Biological Paradigms Affecting the Radiotherapy Outcome. So we are lucky that today we have the Professor Hossein Mozdarani. So before going to introduce himself, I would like to share one slide only. So about the radiation therapy, radiobiology. So as we know, the radiobiology is a branch of science which includes the basic principles of physics and biology, and it is concerned about the ionizing radiation on the living and biological organisms. So the biological effect of radiation results mainly damage to the DNA. However, the therapeutic gain factor is not always achieved because of the different normal structure in the tumor tissue and their response to the radiation. And this is because oxygen level and temperature is not always the same. It also depends on the type of cell population, radiation quality, as well as the capacity of cell repair. So these biological factors which play an important role in radiotherapy outcome will be thoroughly discussed in the today's presentation. So now I am going to introduce the today's presenter, Professor Hossein Mozdarani. So Professor Hossein Mozdarani studied BSc in radiology from Iran University in 1983. Then he completed in MSc in radiation biology and PhD in medical cytogenetics in 1989 from St. Andrews University of UK. He currently holds the position of Professor of the Medical Genetics Department at the Faculty of Medical Science of Tarbiyat Modern University from Iran, where he has been contributing for more than 30 years. He has supervised over 30 PhD and over 130 MSc theses. He has published more than 320 peer-reviewed papers in well-reputed national and international journals, and editor or translator of 21 academic books. He is a leading pioneer in radiobiology and radiation genetic toxicology in Iran, and has a broad background in genetic toxicology of ionizing radiation, as well as chemical and environmental factors affecting human health. He introduced a new class of chemicals as radiation countermeasure, which may be beneficial for the cancer victims. He is also founder, editor-in-chief of the International Journal of Radiation Research and editorial board member of several international national scientific journals. He has been scientific committee member of the AFOMP since 2009. He established a radiogenomic research group in the Iran for the first in 2018. His research interests includes radiation toxigenomics, radiation and chemical carcinogenesis, chemical protection against oxidative stress, DNA damage and DNA repair, translational genomic instability and chromosomal instability syndromes. Now I would like to invite Professor Hossein Mozdarani to deliver his presentation.
Okay, thank you very much.
I will share.
Professor, we have one more information to tell you, that because of technical error, Professor is not able to share their screen. So Professor Arun will share the screen, and Professor Mozdarani will explain the content. Professor Arun, thank you so much.
Thank you. Ladies and gentlemen, I also welcome you to the AFOMP 11th monthly webinar. First of all, I should thank the organizers of this webinar, Professor Chogol, Professor Ajir, Dr. Chai, Dr. Jean, and others involved in arrangement of this webinar. And also I thank the moderator of this webinar, Dr. Akhtaruzzaman, and also Dr. Verma for her help and assistance. Our topic of discussion today is about biological paradigms affecting radiotherapy outcome. What we know that the aim of radiotherapy, please, next slide, please. The aim of radiotherapy is either radical treatment of tumors or palliative radiotherapy. Our main focus is about the radical treatment of tumors. For radical treatment, we need to have therapeutic gain factor. Therapeutic gain factor is defined as the effect of radiation on tumor cells should be more than the effect of radiation on normal cells. It always should be more than one. But therapeutic gain factor is not always achieved. As you see, we can to have therapeutic gain factor or therapeutic ratio as the tumor control probability versus normal tissue complication probability, TCP or NTCP. Whatever the difference between these two curves is higher, the tumor control is better, and the therapeutic ratio is would be also better. Next slide, please. Uh, TGF or therapeutic gain factor is not always achieved because of different cellular structure of normal and tumor tissues and the response to radiation. Here, for example, we have muscle cells. It's a normal tissue. All cells are similar. Non-proliferating cells or quiescent cells. But we have normal tissue like bone marrow cells or like here skin cells that they have some proliferating cells and some functional cells. But all these cells from quiescent cells or proliferating cells in normal tissue have similar oxygen tension, have normal repair phenomenon and mechanisms operating when a damage occurs in the cells. But in the tumor structure, we have different cells. In the tumors are composed of necrotic cells in the middle, hypoxic cells around the necrotic cells between necrotic and oxic cells, and oxic or aerated cells that, for example, are proliferating. We consider most of the cells in a tumor proliferating except the core of the tumor that are inactivated cells or dead cells. There are other things in the tumor structure for cancer stem cells that might affect the radiotherapy outcome, and different microenvironment of the tumor is different from the normal tissue because of different of pH in this condition. Okay, next slide, please. Cells undergo different pathways after irradiations. Cells also are biological effects of cells at atomic level is either direct or indirect. With low electric radiation, we have mainly indirect effect of radiation because of the free radical oxygens. Here we have, for example, water radiolysis that leads to free radicals, and these free radicals affects the cells or biomolecules. All biological effects on the cells are biological effects of the cells are random process. Next slide, please. From ionization events after impartation of energy and the effect of free radicals on biomolecules and the cells ultimate biological effects is ultimate biological effect is indicating the cell's fate.
Professor, you have to unmute. You got muted. We are not able to listen.
I think because of some connectivity issue, he has we lost his signals. He's not there in the meeting. I think he'll join again.
But I can see him as new.
He's joining again.
Oh no, he's not here. Maybe the connectivity issue.
He's not there in the meeting, I guess. Somehow we lost his connectivity with us.
So dear participants, wait a bit. We have the connection issue, network issue. Please be patient.
Professor Chogley, can I request you to continue with some introductory thing till the time Professor joins us?
Okay. Very difficult because his slides. I think we'll wait for some time. Is he not in waiting?
No, we can't see him.
No, but
Yeah, not yet. So he's not there. So I think we should. It's quite a time. Yeah, he has come. Yep, he's joining us.
Sorry, I have lost my connection.
okay.
Okay.
So, the slide or should I go ahead?
Okay. We have all the processes in the tissue as random process, from impartation of energy to DNA damage to sorry, can you hear me?
Yes, we can hear you,
okay.
Professor. Please go ahead.
Okay. and all the processes are random, leading to biological effects at cellular level. Can I have my next slide, please? Cells undergo after different pathways after irradiations. Repair or survival, permanent cell cycle arrest, which leads to cell death, apoptosis, programmed cell death, or mitotic death or mitotic catastrophe. All of these happens both in normal tissue and in tumor tissue when we irradiate tumor sites. Can I have next slide, please? Normal tissue radiology depends on cell population type, whether they have stem cells or proliferating cells, radiation quality, whether high LT or low LT, oxygen level and temperature, cellular repair capacity. As we know, many cells at different ages have different repair capacity. Therefore, these are affecting on normal cells. Next slide, please. As you see here, for example, for high LT radiation, we have a steep survival curve, and for low LT radiation, we have a shoulder survival curve that shows that for low LT radiation, cells can repair themselves and resist to the radiation we deliver. And also for the effect of oxygen in oxygenated or aerated cells, we have a steep survival curve, but for hypoxic cells or anoxic cells, we have a shouldered and with less steep survival curve that shows the difference between the effect of radiation on aerated cells or anoxic cells. Next slide, please. But in tumor radiobiology is different because the tumor consists of different types of cells. Also, we consider all tumor proliferating cells. You see here all these green layers are proliferating cells, and in the middle, in purple, core of the tumor is necrotic cells, and there is one or two layer of hypoxic cells situated between the aerated and necrotic cells. This one is hypoxic cells. Therefore, when we irradiate the tumor, different response will be expected because aerated cells are radiosensitive cells and they will be killed, but the hypoxic cells will resist to the radiation and will resist and will not die with the, for example, 1 or 2 or 3 gray we deliver in every fraction. Therefore, we have different cellular response for tumor cells. And also for necrotic cells, they are anoxic cells. They have no nutrients, therefore they are dead cells in the middle. If we have a tumor with 10 centimeter of diameter, the tumor, the proliferating part of the cells are only 200 microns, and the surrounding hypoxic cells are only 1 or 2 layers. Therefore, most of the tumor cells are necrotic, and we have to combat with only 200 microns of the proliferating tumor cells. Can I have next slide, please? For different structure of normal tissue and tumor tissue, it was shown that fractionation is a better method to produce therapeutic gain factor. we do fractionation because of the differences in response of normal and tumor tissue to radiation. It was found that fractionated radiation is better for tumor control over the single radiation dose. So fractionation is in routine use in radiotherapy. There are different types of fractionation. For example, conventional fractionation, 2 gray per day for 5 days a week. Hyperfractionation, 2 fraction per day. Accelerated fractionation, 3 fraction per day. And hypofractionation with higher radiation dose and lower fractionation and with higher fractionated dose and 4 fraction per weeks for 3 weeks. The rationale for every of these fractionation modality is shown that, for example, we do hyperfractionation to spares late responding tissue. We do accelerated fractionation to overcome accelerated tumor repopulation and else. Can I have next slides? Whatever we do with the fractionation modalities, either conventional fractionation or hypo or hyperfraction or accelerated fractionation, after the first dose delivered to the tumors, we have at least five mechanisms that will be activated in tumor population. The first one is repair of sublethal damage. I will discuss in the next slide in more detail about repair. And we have repopulation, replacement of dead cells by proliferating cells, redistribution, distribution of cells in the cell cycle, and reoxygenation, also radiosensitivity of the tumors. Can I have the next slide, please? After irradiation, we will have different types of damage occurred at the DNA level. Is base damage, nucleotide damage, single strand break, double strand breaks. Double strand breaks is the main damage that leads to the cell killing. In fact, this is the killing effect of the radiation at the molecular level. But base damage, nucleotide damage, single strand breaks, these are sublethal damage that cells can effectively repair all of these damages. However, we have extensive number of base damage after 1 or 2 gray irradiation. We have for 1 gray 100,000 single strand breaks, and we will have about 50 double strand breaks. However, after we study the cells as mitosis, after inducing all these damages, we only see 1 or 2 chromosomal break that may lead to mitotic death of the cell cycle. It means that the cells have the capacity to repair most of these damages. Can I have the next slide, please? The repair process will start after sensing DNA damage with various sensors, signal transductions, effectors, and these sensing sensors, in fact, leads to the various types of DNA damage will operate at the cell at the cellular level. Can I have the next slide, please? Okay. Can I have the next slide, please? DNA damage is next one, please. Repaired by different mechanisms. for single strand breaks and base damage, we have excision repair mechanisms, nucleotide excision repair mechanisms, or base damage excision repair mechanisms, which remove more than 90% of the damages within 10 minutes after induction of the damage. But for double strand breaks, we have non-homologous enjoining, which is a error prone mechanism for DNA double strand break repair, and homologous recombination, which leads to error-free repair of double strand breaks. All these operates at a molecular level to make the cell be alive. Can I have the next slide, please? Okay. Okay. After repair of the induced damages, some cells in the tumor tissue, even during irradiation, can repopulate, can reproduce themselves and produce new cells to replace the lost cells after irradiation. We know that some cells are lost because of the oxygen tension, because of the non-repairable DNA damages, but for the replacement of the cell loss, cells will undergo repopulation and will reproduce themselves. This is very important for late responding tissue because of the overall treatment time. And that's why we need to have at least 6 hours for each fraction. For even for accelerated fractionation, we need to have at least 6 hours between each fraction to prevent repopulation of the cells and also to prevent repair of the DNA double strand breaks, which leads to cell killing. Can I have the next slide? The other things will affect after irradiation of the tumor cells is the reassortment. We know cells are at populating cells or are at different stages of the cell cycle. For example, G1S, DNA synthesis, G2, and mitosis. And for example, cells stay at G1 for longer time than G2. For example, for cells with overall cell cycle time of 24 hours, cells stay at G1 at about 11 hours, 6 hours at S phase, or G2 for 4 hours. Therefore, all the cells are not synchronized in a tumor population. Therefore, we have a mixture of cells at different cell cycles. Irradiation leads to reassortments. It means that when irradiation occurs, cells will stop at G2 cell cycle. G2 has a checkpoint between G2 and mitosis, and this checkpoint prevents entering G2 cells to mitosis. Therefore, cells will block at this stage at G2, but it will allow cells to move in the cell cycle to reach to the cell cycle, to G2 cell cycle. Blocking cells in the radiosensitive G2 phase is one of the major effects of fractionated radiation dose, allowing cells to redistribute enter cells into this radiosensitive phase. If we deliver radiation at this stage, therefore we will have more radiation effect. The other main phenomenon will occur after irradiation is reoxygenation of hypoxic cells. As said, the diameter or thickness of oxygenated cells, oxygenated proliferated cells is about 200 micrometer. This leads to some cells not receiving enough oxygen or nutrients. Therefore, these cells will be hypoxic. These are not dead cells like anoxic cells and not dividing cells like aerated cells, but they are colonogenic cells that when the condition is or the situation is suitable, they can regenerate and repopulate and produce new cells. These are colonogenic cells. When some cells after radiation, can I have the previous slide, please? Previous one. Yes, thank you. When we irradiate cells, some of these cells, because of the high oxygen tension, will die, and more oxygen will reach to these hypoxic cells. Therefore, these cells will be oxygenated again. This process is called reoxygenation. Presence of hypoxic cells is one of the main limitation in or main limiting factor in appropriate radiotherapy, because after the completion of the radiotherapy course, even less than 1% of these cells are present or not affected by radiation. They will make the tumor recurrence at the site of tumor. Next slide, please. Radiosensitivity is another issue in radiotherapy. As Bergonier and Tribundo indicated that some cellular systems are more radiosensitive than other cellular systems. For example, bone marrow cells, because of the presence of stem cells and proliferating cells, are more radiosensitive than skin, which have only one layer of self-renewal cells, and skin cells are more radiosensitive than neurons or muscle cells, because these are composed of non-proliferating cells. Therefore, whenever the cellular system is composed of more or highly repopulating cell, therefore it will be more radiosensitive. This is the case for tumors as well. Malignant tissues are more radiosensitive than normal tissue as they have greater reproductive capacity. But all tumors are not equally radiosensitive, as their dividing rate and the fraction of cells present in the resting phase of the cell cycle are varied. Radiosensitivity of tumors or normal tissue is also influenced by many other factors such as LET, dose rate, oxygen concentration, cell cycle status, repair capacity, and etc. Can I have the next slide? Thank you. The key point in fractionated radiotherapy from the point of view of this phenomenon we discussed shows that the timing in dose delivery is very important. We should have a very good timing or appropriate timing for repair of normal tissue, to allow normal tissue repair, not tumor cells. We should have appropriate timing to have appropriate oxygenation of hypoxic cells. We should have appropriate time for reassortment or redistribution of dividing cells to accumulate in radiosensitive phase of the cell cycle at G2 phase of the cell cycle. we should have timing to not to allow enough time for repopulation. And yes, we don't have, we shouldn't allow enough time for repopulation to replace the dead cells by new generated cells. Okay. These were not the only biological phenomenon that affect radiotherapy outcome. At the cellular level, we, as said, we have genetic alterations at DNA level. In this genetic alteration at DNA level translates into gene mutation, microsatellite instability, cell cycle checkpoint perturbance, chromosomal instability. All these effects will lead to genome instability of the cells at tumors, leading to cell death, chromosomal aberration, gene mutations, apoptosis, and cell cycle arrest, etc. Can I have the next slide? This genome instability at the tumor cell population will lead to different phenomenon. For example, adaptive response. Because some cells receive more radiation, will be killed, and some less, or some not hit at all. Even we focus the radiation on the target volume. Some cells will receive less than radiation, less radiation than suitable to kill the cells. This will lead to adaptation or adaptive response that will make the cell more resistant to the next radiation. The genomic instability also will induce bystander effect, which also has its own consequence in tumor cells, and also genetic sensitivity of the tumor cells. I will briefly explain this phenomenon, how to affect the effect of radiation on tumor population. Can I have the next slide, please? Intrinsic radiosensitivity is very important for tumor control, because if intrinsic radiosensitivity exists, tumor normal tissue complication will be more than in individuals with normal radiosensitivity. Also, radiosensitivity is a controversial issue. We really don't know who is a radiosensitive and what is radiosensitivity associated with. But literature says that about 10% of apparently normal individuals are radiosensitive. We say radiosensitive means intrinsic radiosensitivity. This has been shown in different genetic conditions like ataxia telangiectasia, Fanconi anemia, xeroderma pigmentosum, Bloom syndrome. These genetic conditions show higher radiosensitivity. Intrinsic radiosensitivity was first seen in ataxia telangiectasia. The patients that are more prone to cancers, these patients could not tolerate normal radiotherapy regimens compared to other cancer patients. These patients show 2 to 3 times more radiosensitive in normal tissue because of intrinsic radiosensitivity. Can I have the next slide, please? This is the example for radiosensitivity in cancer patients. For example, this is our works for breast cancer patients. With a simple G2 assay, irradiating cells at G2 cell cycle irradiated not cancer cells, but normal cells derived from cancer patients, breast cancer patients, irradiated in vitro and studied at mitosis, showed that higher frequency of chromosomal aberrations, about 50% of breast cancer patients show higher radiosensitivity compared to other cancer types or control individuals. It is a predictive assay for radiosensitivity. Also, we use a comet assay to show why these higher chromosomal aberrations is induced in breast cancer patients' lymphocytes. We saw that the repair mechanism is different in breast cancer patients versus normal cancer patients. We see after 24 hours, repair of about 30% of DNA damage is not repaired in normal cells. Okay, implication of radiosensitivity in radiotherapy. Patients with higher radiosensitivity, can I have the previous slide, thank you. Patients with higher radiosensitivity cannot tolerate chemo and radiotherapy, conventional chemo and radiotherapy as normal individual. And also, we expect higher frequencies of secondary cancer after radiotherapy in these patients because more unrepaired damage will persist in their tissues. Radio adaptation is another features after receiving radiation. Radio adaptation is mild stress that lead to cells can tolerate the next stress, next severe stress. Adaptive response occurs at very low doses. It might happen in tumor tissue, and therefore this very low dose will make the cells more resistant to radiation. This is an example of how adaptive response is explained. For example, if we say control cells, we score the chromosomal breaks is near zero, and if we give these cells, these normal cells, 10 centigrade, we will have the same number of chromosomal aberration as controls. If we give the cells 1 gray radiation, we will have 100% chromosome breaks. But if we give the cells 100 centigrade before irradiation of 1 gray, then we will have much less chromosomal breaks after 1 gray irradiation. This is the concept of radio adaptation. First, it considers as radiation hormesis. Can I have the next slide, please? Radio adaptation impact on radiotherapy because intervene with cell killing effect of radiation and chemotherapeutics leading to lower therapeutic efficiency. The reason for induction of radioadaptation might be activation of repair process. However, some believes that no repair might be more beneficial because damaged cells might die and replaced with normal cells. If cells are alive after misrepair, this will lead to secondary cancers and or recurrence of cancers because of the cells is alive with altered genetic material. Can I have the next slide? Another phenomenon we discuss about is bystander effect. Till a few years ago, the concept of radiobiology was based on the target effect of radiation. In fact, we in that concept, a cell heated will have will show its consequences. It will have altered cellular damages or DNA damages, or it will be killed, etc. But in bystander effects, the heated cells is not the only cells that will be affected by irradiation, but some other cells distant from the heated cells will be affected by the effect of the heated cells, non-targeted cells. There are growing evidence that shown that multicellular response can be induced in the non-irradiated cells that are in the vicinity of the cells irradiated by low and high LET radiation. This is the impact of how the concept of bystander effect. Bystander effect, we will see the effect in non-targeted, non-heated cells around the cells via different mechanisms, by signal transduction, or by gap junction between the cells. This effect will be transmitted. Bystander effect will be induced in different forms, increase in DNA damage or proteins, decrease in DNA damage, or increase in reactive oxygen species, cell death, cell proliferation, mutation, chromosome aberration, chromosome instability. In fact, bystander effect is a double sword در واقع after irradiation, which happens at cellular level. Implication of radiation-induced bystander effects include induction of second cancers, induction of genomic instability and delayed or immediate mutations in areas not receiving a direct radiation dose. The promitogenic effects are observed in some systems, probably influenced by result of radiotherapy through an increase in survival of targeted cells due to bystander adaptation effects, leading to increased risk of tumor regrowth and modulation of potential targets in radiotherapy. It may this bystander effect might also induce bystander adaptation effects in tumor cells. The last effects we see at the cellular level, which is considered as very beneficial effects of radiotherapy, is abscopal effect, which is also called the distant bystander effect. We see bystander effect in the cells at the vicinity of the target cells, but we see abscopal effects in other sites of the body irradiated. For example, if you have multiple myeloma and you, for example, irradiate one site of one of the myeloma in the body of the patients, the other site of the body which is not irradiated will shrink as well as the irradiated tumors. This is called bystander effect, which is a very beneficial effect of radiotherapy. It is sometimes observed, and it is there are different case report about this. This has been shown for most observable tumors, not observable tumors, that we can see the effect of radiation without with eyes without any problem. And therefore, this is another a beneficial effect of radiotherapy. If happens, it will be very good, especially for metastatic tumors. Therefore, if you irradiate the actual tumor sites, then it might affect the metastasis in other sites of the body and will kill the metastatic cells in the body with one radiation. Can I have the next slide, please? These are different case reports from 1969. There are increasing case reports about these abscopal effects. We have done experimentally on mice, and we have seen with different breast cancer tumors that we can have abscopal effect in different parts of the body of the mice. We did this experimentally, but there are few experimental evidence for abscopal effect. Can I have the next slide, please? How abscopal effect operates is not really known. It might be immune mediated. It might be mediated by p53. It might be involved in DNA repair mechanism and some other epigenetic mechanisms, for example, DNA methylation, small RNAs, and long non-coding RNAs, and several mechanisms are proposed, but we actually don't know how this effect is observed after radiotherapy of main tumor and its effect on the other tumors in other parts of the body, not in the field of radiation. Can I have the next slide, please? In conclusion, daily fractionated low dose results in activation of various cellular mechanisms leading to decreased cell killing effects of radiation. Timing in dose deliveries in fractionated radiotherapy is critical for radiotherapy efficacy. Radiosensitivity, adaptive response, bystander effects are key players in radiation-induced cellular alteration leading to radio-resistant and secondary cancer. All these phenomena apparently involve the same genetic background and signaling pathways, mainly genomic instability, and its player bystander effects indicate a non-linearity into the low dose area we will have in non-homogeneous irradiation in the tumor environment. Thank you very much for your patience and sorry about the inconveniences about the communication I had. Thank you very much. If there is any question, I am prepared to answer.
Dr. Akhtar?
Dr. Akhtar?
Okay. So just I am looking at a question. How many fractions and dose per fraction prescribed dose are defined for accelerated radiotherapy?
3 fractions per day, but for yes, 3 fractions per day, and the condition is you should have at least 6 hours time between each fraction.
Okay.
This is the minimum time for repair. This is the minimum repair time for to allow normal tissue restore their damages.
Okay. So there is another question. What condition we should use BED and what condition we should use EQ2? Did all tumors have alpha beta equal to 10?
No, as I said, different tumors have different radiosensitivity. Therefore, they don't have similar alpha beta ratio, you know, and we should determine alpha beta ratio for each tumors.
Okay. So always, yeah, because different kind of tumors have got different alpha beta. Also, the early reaction, the late reaction alpha beta will be different.
Yes.
And therefore, the BDs will be different for different kind of tumors. And
that's right. Yes.
equal BD, then number of fractions or dose per fractions has to be adjusted. So these were some of the questions from the participant. So once again, I thank Professor Hossein for giving a wonderful talk on importance of radiobiology into the radiotherapy, because there is a basic thing how you are going, because it's not the physical dose, it's a biological dose, what we say for the cure or the reaction of the radiation which we are going to give it. So once again, I thank Professor. Thanks to
Thank you very much.
Mohammad Akhtar for the moderation of these things. And thanks the participant for participating. We have next webinar on 6th May 2021, and this will be given by Dr. Hyun Tai Chung from South Korea. And the title of the talk is Basic Principles, Dose Planning, Advantages and Quality Assurance of Gamma Knife Radiosurgery. And I hope all you will participate and get advantage of the AFOMP monthly webinars. As you will go and visit the AFOMP website, you will find the schedule up to June 2021. Already we have decided to continue this monthly webinar, and we have prepared the schedule for July to December. In a day or two, it will be available on the AFOMP website. The recording of this webinar will also be available on the AFOMP website. This is a CPD accredited, and the attendees can get 2 CME points. So the link has been given, so you can register on that link, send the email. Our PRC chair will send you the certificate of the attendance. With this words, sorry for some inconvenience. These are the technical beyond control. Thanking all the AFOM office bearers, Dr. Rajni Verma, who is taking a lot of efforts to coordinate, communicate you, and overseeing all this success of the webinar. Thanking with all of you. Thanking the orator of this today's AFOM. I will close this session with these words and looking forward that you will join on 6th May 2021 for the next webinar of the AFOM. Thank you all. Have a nice day. Stay safe.
Thank you very much.